Our next speaker is Dr. Cristina Alvira, an assistant professor in the Department of Pediatrics, and she'll be discussing the developmental regulations of pulmonary angiogenesis by the lung.
Great. Thank you so much for this opportunity to present our work. Okay. So it's not always well appreciated, but in contrast to many other organs which complete their development very early in pregnancy, a significant component of lung development actually happens in late pregnancy and during the first decade of postnatal life. And this occurs during the alveolar stage of development, which begins only just before term birth and probably extends for the first 10 years. And it's during this transition from the saccular to the alveolar stage of development where these larger terminal air spaces are subdivided by a process of secondary septation, resulting in millions of smaller alveoli, and this dramatically increases the gas exchange surface area of the lung. However, the completion of the lung's development late in pregnancy and in postnatal life really renders the immature lung highly susceptible to injuries that can disrupt this developmental program. And this includes numerous postnatal injuries that I encounter as a pediatric critical care physician, such as mechanical ventilation, high amounts of oxygen, infections, and corticosteroids, as well as antenatal exposures that are happening well before alveolarization even begins, such as intrauterine infection or intrauterine growth restriction, which also has been shown to disrupt alveolarization. And really much of what we know about how alveolarization occurs has been learned by studying the disrupted lung development in babies who are born prematurely who develop a chronic lung disease called bronchopulmonary dysplasia, or BPD. And recent evidence suggests that the pulmonary vasculature is really key to this process of alveolarization. And in this vascular hypothesis, which was really based on, in large part, work done by Steve Abman at the University of Colorado in Denver and Bernard Thibault at the Ottawa Research Institute. And it was based upon the observations that if you looked at the lungs of babies who ended up dying from PPD, these larger and fewer air spaces, which we call alveolar simplification, was associated with an overall decrease in the amount of angiogenic factors in the lung. And in animal models, if you directly block angiogenesis, you induce the same alveolar simplification. Yet if you use strategies to replace angiogenic factors in those same animal models, you can preserve or enhance alveolar development during times of injury. So this really led us to believe that if we could develop a deeper understanding of the molecular control of pulmonary angiogenesis during development, that we might be able to leverage that knowledge into strategies that could be useful for enhancing or promoting lung growth in premature babies and even older infants and children. So we became very interested in the role of a specific signaling pathway in regulating pulmonary angiogenesis during late lung development. And this signaling pathway is the NFKB pathway, and as many of you know, this pathway is really a family of transcription factors, and these NFKB molecules, which exist as dimers, are really expressed in all cells, but held in an inactive state in the cytoplasm. And they're activated by a complex of kinases, which you see here, consisting of the kinases IKK alpha and beta, which end up phosphorylating an inhibitory protein on the NFKB molecule, which allows it to translocate into the nucleus where it regulates gene expression. And so because the primary activation step for this pathway is the nuclear translocation of the molecule, this gave us kind of a quick and dirty way in which we could quickly analyze how much active NF-kappa B there was in lungs at different stages of development by doing confocal immunofluorescent microscopy to detect active nuclear NF-kappa B molecules. And so as you can see here, we first look at lung sections taken from mice at postnatal day 6, and mice actually start alveolarization at postnatal day 5, so they represent a really nice model for premature BPD or late lung development. And what we found was that when we looked for the expression of the NF-kappa B dimer P65, we saw a lot of nuclear staining in the lung at this stage in development. And this was in contrast to the adult lung, where the NF-kappa B expression was primarily cytoplasmic. And as you can see, most of the nuclei are blue with an absence of active NF-kappa B molecules. And when we did studies where we actually isolated primary pulmonary endothelial cells from the lungs at different stages of development, we found a similar effect. We found that the NF-kappa activation in pulmonary endothelium was dynamic during development, where we had peak activation at this early alveolar stage. And by late alveolarization, where angiogenesis is slowing and there's a period of microvascular maturation, the pathway seemed to be diminishing. And in cells that were obtained from the adult lung, we actually saw no activation of the pathway. And importantly, when we looked as well, these early alveolar pulmonary endothelial cells phenotypically were different than the adult cells. They proliferated faster, they migrated more quickly, they survived better than the adult cells. So as early proof of concept that this pathway seemed to be important at this stage in lung development, we did experiments where we just treated mice at early alveolarization with a global pharmacologic inhibitor of the NF-kappa B pathway. And what we found was 24 hours later, we had a marked impairment in lung development. We had drastically larger airspace size, characteristic of the alveolar simplification that we see in patients with BPD. In contrast, the same treatment to adult mice had absolutely no effect. And more recently, we've created a mouse model where we could induce endothelial-specific deletion of the main NF-kappa B activating kinase, IKK-beta. And when we induce IKK-beta deletion right before alveolarization, we find that that is sufficient to induce airspace dilation, creating these areas of emphysema within the lung. And moreover, the mice that were lacking IKK-beta and endothelial cells had a really marked impairment in alveolarization when exposed to chronic hyperoxia, which is a very standard injury model to impair alveolarization. So now that we knew that the pathway was important, we really wanted to understand what was controlling this temporal specific activation of NF-kappa B in this discrete time point in development. And we thought that it could be either one of two things. Either there was something intrinsically different about the two cells, the early alveolar pulmonary endothelial cell versus the adult, or it was that the cells were relatively similar, but they were responding to differences in the lung microenvironment. So to test this hypothesis, we did a relatively simple experiment where we just created an organ culture model, and we collected the secreted factors from the lung at different stages of development. And then we added them to the adult cells where NF-kappa B is inactive, and to assess whether or not they could activate the pathway. And what we found was that secreted factors from the early alveolar lung conditioned media, which you see here, the yellow bar in this image here, actually very robustly activated the NF-kappa B pathway in the adult pulmonary endothelial cells. However, secreted factors taken just 10 days later, again at a stage in lung development in the mice where angiogenesis is decreasing, really had only a modest effect at activating this pathway. And we saw no activation when we used secreted factors that were collected from the adult lung. But what was more interesting was actually that these secreted factors seemed to change the phenotype of the adult cells. So when we did scratch assays to assess adult pulmonary endothelial cell migration, we found that the addition of this early alveolar lung conditioned media was as effective as 5% serum in inducing the adult cells to migrate. And again, we saw very little effect when we used the lung conditioned media that was collected from the adult lung. So then we wanted to know, well, what is it in the what is it in the lung conditioned media that was mediating that effect? So we did proteomic analysis where we compared the proteins expressed in the three media by labeling the proteins in each of the secretomes with a single fluorochrome and then running all the proteins together on a single gel and then comparing the signals from any two media against one another. And we were able to narrow down from hundreds of differentially regulated proteins by first looking for factors that were only present in the two alveolar secretomes and not at all expressed in the adult secretome. And then they also had to be much more highly expressed at the early alveolar stage than the late alveolar stage. And then out of that group, we identified the top 30 via mass spec and did some screening analysis to look for proteins that seemed like they might have an effect on either angiogenesis and NF-kappa B activation. And we ended up focusing our efforts on one protein, which is shown here. And this protein is called TGF-beta induced protein. So not TGF-beta 1, but TGF-beta induced protein. And we first confirmed, both in the lung-conditioned media as well as in lung tissue obtained from mice at different stages of development, that there was high expression of TGFBI, the early alveolar state, and it really disappeared by early in the adult lung. And when we did immunofluorescence staining, again, looking for the expression of TGFBI in situ, we found in the early alveolar lung there were cells that were just very intensely positive for TGFBI, and really a complete absence of TGFBI staining in the adult lung. And on higher magnification, we saw that a lot of times these positive cells were at the tips of the secondary crests, which are dividing those larger saccules into smaller alveoli, and these tend to be locations where we find myofibroblasts. And so more recent new data from our lab using single-cell techniques to try and look at the gene expression of vascular cells at different stages of development found, really confirmed, I think, our findings on immunofluorescence, where we have high expression of TGFBI in a myofibroblast population in the early alveolar lung, as well as expression of TGFBI in a number of macrophage populations. So we then wanted to know, well, how much of the effect of the early alveolar lung conditioned media and mediating NFKB activation and promoting migration was really related to TGFBI. So we did experiments where we added either isotype control antibodies or anti-TGFBI neutralizing antibodies to the conditioned media, and we showed that while the conditioned media containing control antibodies could still robustly activate NF-kappa B. This was blunted by the addition of the anti-TGFBI neutralizing antibodies. And similarly with migration, we were able to completely block the pro-migratory effect of the lung-conditioned media by adding the anti-TGFBI neutralizing antibodies. We then did experiments where we looked at the role of the recombinant TGFBI alone and its ability to induce some of these effects. And these studies were actually done in collaboration with Dr. Heilshorn's group. Sarah Heilshorn is an engineer here at Stanford, and this was part of the MCHRI Transdisciplinary Initiative program. And they were able to create for us microfluidic chemotaxis assays where there was a source for the TGFBI on the right and a sink on the left, which created a stable linear gradient of TGFBI within a cell chamber. And then the endothelial cells were grown in this chamber and monitored by live video microscopy and tracked in order to create these directional histograms where the size of each wed represents the number of cells migrating within that specific angular trajectory. And so we were able to show in the starvation media that there was a non-significant trend drift kind of to the left. And this was in contrast to our positive control where we had directed migration towards the source. We saw similar directed migration when we used either the early alveolar lung conditioned media or the recombinant TGFBI alone. We also showed that the recombinant TGFBI was able to induce NF-kappa B activation both by immunofluorescence and also looking at the amount of NF-kappa B DNA binding by EMSA. And moreover, the ability of TGFBI to induce cell migration really appears to be NF-kappa B dependent because when we either treated the cells with that pharmacologic inhibitor BAE, or if we used our cells that were lacking the IKK beta activating kinase, we lost the TGFBI-mediated migration. And then finally, we collaborated with Simon Conway's group in the Indiana University, and he had recently created a TGFBI global knockout mouse, and we found that these loss of TGFBI impairs alveolarization under baseline conditions, and this is associated with an overall reduction in the pulmonary vascular density in the lung, as well as really dramatically worsening or exaggerating the effects of chronic hyperoxia, both on the alveolarization as well as on pulmonary vascularization. So in summary, growth of the pulmonary vasculature by angiogenesis is really essential for alveolarization. And the angiogenic phenotype of the pulmonary endothelial cells is developmentally regulated, and NF-kappa B seems to be one pathway that's very important in mediating this phenotypic switch. And unique factors present in the early alveolar lung microenvironment seem to be very important in modulating the angiogenic phenotype of the pulmonary endothelial cells. And I would just like to acknowledge everyone, past and present members of the group who contributed to this work, as well as all of our great funding sources, and most specifically the Maternal Child Health Research Institute, just both for the TIP grant and also for the Tasha and John Mortgage Faculty Scholar Award. So the question was about the population of TGFBI expressing cells in the lung. Actually, the TGFBI expressing cells in the lung are not the endothelial cells. So they're the cells present at the tips of the secondary crests, which are typically myofibroblasts, and our single cell lung RNA-seq data also suggests that it's really the myofibroblasts that are kind of signaling to the endothelial cells. So the TGFBI is actually a very interesting molecule because it binds integrins. It also can bind the matrix. So I sort of imagine a scenario. I sort of imagine a scenario where, the tips of the secondary crest are secreting the TGFBI, kind of calling the endothelial cells towards them, because you need to have coordinated development of the vasculature with the developing airway, if that makes sense.
So just to follow up, so during this development, when you get shutdowns, so this is secret protein is shut down or something. Do you think this fibroblast, secreting fibroblast, apoptosis or disappear, or this cell still is there just to shut down this expression of this?
Yeah, so your question is what regulates the TGFBI expression? We don't know, so we're trying to look at that in more detail. I think that's, again, one of the reasons why the single cell work is so helpful, because you realize how much heterogeneity there is, and then we can sort of try and track these cells. We do know that hyperoxia really decreases the expression in the conditioned media, but we don't know if that's because the cells die or if it's not being secreted, or, there's lots of potential mechanisms, so decades of work to continue.
Thank you. And we are all going to stay tuned 10 years from now when you have pilot funds to do the translation work.
Okay, excellent.
And we're taking this into our preterm labor and preterm babies.